Thank you very much. And I want to thank Beth Stevens for inviting me here today to give a talk about one of my favorite subjects, which is the micro world. And today I'm going to talk about the innate sensuality of the micro world. Now, looking at this picture here, you might wonder what this is. This is actually the seed of a strawberry. So this is a strawberry through a microscope cut in section. So to start with, I want to tell you a little bit about myself and sort of the things that I've been doing. I want to start by telling you about some of the websites that you could look up later. When I was a youngster, after college, I was president of this company, Environmental Communications, and environmentalcommunications.info is a website you might want to look at later. This was work I did documenting the zeitgeist in the 1970s in Los Angeles and around the world. After that, I moved to London and got a PhD in developmental biology, and then did research at USC for many years. And then in the 1990s, I started inventing microscopes, and I started a microscope company called Edge. And then I found that microscopes are a wonderful place to do art. So I started doing a lot of art through the microscope, and I'll tell you about that. That'll be the end of the talk. And that's the sandgrains.com website. And I'm currently, I live in Maui, and my current affiliation academically is the University of Hawaii Institute for Astronomy, where I look at moon sand through the microscope, sand that was loaned to us from NASA from the Apollo missions 40 and 45 years ago. So I want to tell you a little bit about what the real point of this talk is. And the real main point of this talk is that microscopes are an incredible instrument for both science and art. And in my life, it's been instrumental in exploring and searching for truth and beauty. That's been really what I've focused on for most of my life. And you might think that, well, I started as a photographer, became a scientist. For me, art and science are the same thing. They're explorations of nature. So as a youngster, this is me, I think, pretty much out of college. That was my first movie camera. It was a Beaulieu 8mm movie camera, and then I got a 16mm movie camera. And I became a photographer. I studied actually psychology. I knew I didn't want to become a psychologist. And I started taking a lot of pictures, and it was a very empowering thing for me to find photography. Photography showed me that you could create a point of view, and it showed me that everybody actually has a point of view. So if I asked you all to take a picture of this room, or of any environment, everyone would take a different picture. Everyone has their own point of view. And by exploring your own point of view through photography, which is a great tool for doing it, you really find out a lot about yourself. So I want to tell you about the power of images. Right about that time that picture was taken of me, NASA was going around the moon. This was the Apollo missions. Apollo 11, 12, 14, 15, and 16 landed on the moon. The earlier Apollo missions went around the moon. And they were far enough from Earth to be able to take whole pictures of the whole Earth. Before we had satellites taking satellite pictures of the Earth, but they never had a picture of the whole Earth. And this image, once it was brought to the consciousness of the world, changed the way people thought of the Earth that we live on. They finally got the idea that we actually all live in a globe floating in space. And a person called Stuart Brand at that time actually had to start a movement to get NASA to take a picture of the whole Earth and release it to people, because they had never taken a picture of the whole Earth. He knew that they could. It took a couple of years for him to finally get NASA to take a picture of the whole Earth. And he did, and he started what was called the Whole Earth Catalog. And that was one of the first pictures that people saw of the whole Earth. And in that catalog, it was a big, thick catalog done in newsprint, and it had all kinds of totally cool things that you could buy. Things like, right here, there's like plans for a ferrocement boat or a motor home, or you could make geodesic domes or inflatables, and everything from Swiss Army knives to everything you can imagine. It was a very, very popular thing. And in those days, catalogs, this is before the internet, so catalogs were kind of, in a way, kind of like the internet. That's how you connected with buying things and finding out what was cool. And so I became the president of a company called Environmental Communications. There were a group of photographers and architects and designers, ecologists, artists. And what we did is we documented the creativity of the 1970s. We were headquartered in Venice, California, and we came out with catalogs. And the catalogs consisted of slides, films, and videotapes and books of art, architecture, environment, ecology, and so forth. And these are some of the covers of some of the catalogs, and we sent them all over the world to universities and libraries, and they would buy them. And some of the stuff and one of the things I did is I bought a motorhome. I bought a GMC motorhome, and I outfitted it with a sunroof so you could get right up high above everything, and it became an incredible platform for taking photography in the urban environment. So we were sort of dedicated to going around the United States. Actually, we're going all around the world. But this became a great platform for photography because you were way up high and you didn't get telephone poles in the way and cars and people, so you could get a really good point of view on the urban environment. This was our headquarters here in Venice in the background. We had the studio downstairs here. We had the upstairs studio here, and we had the roof garden. We built a really cool roof garden up there. So this is some of the stuff. This is back now 45 years ago. These are my partners, my brother David, Roger Webster, Bernard Perloff, Ted Tanaka. That was me in my office. I had a big picture of the moon in back of me. Little did I know, 45 years later, I would actually have bits of the moon in my hand photographing it. These are some of the players. And there was a group of there were 5 partners, about a dozen core people, and about 150 different contributors. So we weren't the only ones doing this at the time. There were lots of other groups like Ant Farm, Chip Lord, who has been associated with the university here for a long time. He was one of our contributors, and we did a lot of work with them. So here's some of the pages in that catalog. So this is we had a couple of pages on ecology. At that time, the word ecology, people didn't even know what it meant. I'm talking about the early 1970s. We were just bringing it to and a lot of people at that time were bringing the problem of pollution to the consciousness of people. And it was photographs like this that really could speak a lot, about photographs more than 1,000 words, and it's really true. And one of the things we did is we realized that there's more art in the environment than there is in museums. Very few people go to museums and see art in the museum. But if you do something like a billboard or neon art or an art from cars, so we had a slide series on custom cars. This is the Dickmobile here. And in those days, by the way, these billboards, they don't do it anymore. They were hand painted. The billboards in Sunset Boulevard, they were like the premier billboards in the world. And they were hand painted. They brought people in from Europe to hand paint these billboards. They were phenomenal, unbelievable. And they were works of art. They were true works of art. And that was one of our slide series, Billboards of LA, neon art. A lot of wall paintings were being done at the time, super graphics. People were expressing themselves in the urban environment so everybody could see it. So when you put a wall painting up there, hundreds of thousands of people see it, maybe even every day. So it sort of blew away the idea of a museum. The urban environment was becoming a museum. We also got very much into architecture because a couple of our partners were architects. This was a time when geodesic domes were becoming very popular, inflatables. People were making inflatables. They were very easy and cheap to make. You could make a structure and do all kinds of stuff with it. It was portable. It was super cool. We had slide series on houses or structures that looked like things. This one here was a slide series done by Tom Sewell, who was one of our contributors. He actually bought that because they were going to demolish it. He bought it and put it on a big truck and moved it to a spot in Venice. And about a week later, it collapsed it had too much termite damage and it actually collapsed. So that was unfortunate. But that was a really interesting slide series. I went to Australia and did a slide series on the Sydney Opera House. Now, you guys have all seen the Sydney Opera House, but I'm not sure if the importance of this building can't be underestimated. This guy, Jorn Utzon, was a young architect from Denmark, and he won the competition in 1958 to do this building. This is way before computers were ever used in architecture. I mean, decades before. Every tile on this thing is different. It's phenomenal. When this was being built, this guy got in a lot of trouble. He was, it was so over budget and so over time. But finally, after it was built, it became it's the icon of Sydney. It's the icon of Australia. And it's one of the most fantastic buildings in the world, really phenomenal. And at the same time that was happening, this guy, Frank Gehry, was in Venice building buildings like this here. In 1970, this was earlier actually, but in 1970, 69, late 60s, 70s, Frank Gehry was building buildings like that. And over time, Frank Gehry's buildings developed into buildings like this, the Disney Center. And this all was really inspired by the Sydney Opera House. Now think of this building here, Frank Gehry's building, without having computers today. And that's what this guy did in the early 60s. Here's a couple more pages from our catalog. Ant Farm had a page. They did a thing called Cadillac Ranch, where they buried a dozen Cadillacs face down in Texas. They did a lot of really super cool stuff. There was another group called Archaeogram, another group called Chrysalis in England. So it was really, this catalog was kind of a nexus of a lot of creative people exploring the zeitgeist of the 1970s. Now, an interesting thing happened is because we had these slide sets all over the world, a couple of years ago, some professors from USC and Columbia University discovered our slide sets in their library. And they thought, oh man, this is pretty cool. So they started putting on these retrospective shows. And there was one in Columbia University, there was one at the Chicago Biennale. This is the one that just closed in LA a couple of weeks ago. And they were really cool. They had all our slides there. Each slide set had a booklet that went with it. These are some of the booklets that were there. There was a bunch of slides. There was rooms on the side that had videos. This is when video was just really happening in the 70s. We were the first to have time-lapse video machines. When there was a big earthquake in '71 in Los Angeles, we were hired by Goodyear Blimp to go up in the Goodyear Blimp and take pictures of the devastation. We even had a room with some actual. This is how we used to look at pictures, by the way. This doesn't happen anymore. But this is how you'd go and you'd have these slide shows, actual slide shows. Now this was a big long. There's a bunch of TVs, monitors put together, and that was a crawling. Those images were crawling along, but they actually looked like slides. So this is us then and now. So that's me. I've become an old man. Ted Tanaka, he's become a very famous architect. He's redoing the LA airport, and he's doing the stations at the LA Underground, mass transit. And Roger Webster became Mona, which is interesting. Now at the time, Roger Webster had one thumb painted red. And it evolved from that to Mona. So that's interesting. So I want to talk now about my early influences. I was a photographer after I came out of college, and I was very much influenced by a lot of the art at the time and a lot of the erotic art. And one of the first things I ever came across were the Kama Sutra temples. Now these things are 1,000 years old, and they have sculptures all around them. And the sculptures portrayed what would be a everyday good life, how you live a good life for yourself. And in part of that living a good life were these sorts of images and these sorts of sculptures. So that's pretty interesting how the mores and morales have changed from then to now. I want to talk a little bit about Georgia O'Keeffe, who was one of the great artists. She was a painter and watercolor artist. She was married to Alfred Stieglitz, who was one of the greatest photographers of the time. And she did these fabulous pictures, I mean, just boldly erotic pictures of flowers. I just want to show you some of these. Georgia O'Keeffe. Georgia O'Keeffe. She was really happening in the 20s and 30s. She was fabulous. Now here was this guy, Aubrey Beardsley. Now look at when he was born in 1872. He died of tuberculosis at the age of 25. He was an author and an illustrator. Now in those days, books were illuminated or illustrated. They didn't really use photography, although photography did exist at that time. They weren't really in books. And he was very much influenced by Japanese woodblock kind of art. And this was some of his illustrations from Salome. And he was really one of the first, this was the sort of before Deco art. He was the beginning of Art Deco, which really flourished 1910, 1920. And he had some pretty much outright erotic things in his art, and I'll get to some of that. But he was a really, really inspiring artist at the time. And unfortunately died quite young. Can't imagine what his career would have been. So here's some of he got into a lot of penis stuff as well. Just to show you a little bit of that. Now another person who really influenced me a lot was Edward Weston. He was a very, very well-known photographer. And he did a series of photographs for years of bell peppers. Now in those days, there were 8x10 cameras. And what you'd do is you'd put a sheet of film, 8x10, in the back of the camera, and there was a slide to keep it from being exposed. And you'd see it, and you'd see the picture upside down. And it's interesting, so when you were a photographer in those days, your sense of competition, it had to work upside down and right side up. I had a 4x5 when I was a young photographer. I had a 4x5 a plate film camera, and it was really interesting because you get this fabulous resolution, but you had to see the picture upside down. So we did this series of bell peppers, and one was more sensual than the other. He did this for years. And he would light them just right. And his bell pepper series was one of his most famous things that he did. So these were all, by the way, super high resolution images that he'd make these big, beautiful museum prints out of. And he'd do these fabulous shows with these big, huge, beautiful prints. Now this is a friend of mine who was around in the environmental communications days. I just saw him the other day at my opening in LA, Guy Dill. And Guy Dill does these wonderful, sensual sculptures. Here's an example of one. And what he does is he explores shapes that are innately erotic and innately sensual. So this is your assignment for this week. What I'd like you to do is do something like a little report on what is an innately sensual shape, pattern, shapes, patterns, textures, and why are things innately sensual or innately erotic. Maybe write something less than the page and find examples, either create an examples or find examples, either paintings, drawings, photography, montage, and see if you can find shapes, textures, and patterns that just have this innate sensuality to it. So that's what my talk is really about, and I'm going to show you how the microscope is one of those things that really can allow you to do that. So the microscope allows you to see hidden worlds that you could never ever see without having this ability to see tiny things that our eyes simply cannot see. Now the first lenses were made by a drop of water. And the interesting thing is the first spectacles were around about the end of the 1200s in Italy. Spectacles were quite common, but it wasn't until 1600 that they invented the microscope. Now, microscopes and telescopes were both invented at 1600 in Holland. And all it really was, if you think about it, it's taking two lenses and holding them up like that. And depending on where you put your eye and what the focal length of the lenses are, it's a telescope or a microscope. Now, the deal is, how come it took from 1280 to 1600 for somebody to do that? Well, I'm sure people did do that, but they didn't do anything about it. And this is one of the main things in life. You come up with ideas, and oh, that's cool, and then, the next day it's gone. It's people who really have the passion to follow through and do something about it that make real changes in the world, that create and invent new things. So Hans Lippershey was the one who invented the first telescope. This is him. That's a replica of his first telescope. Microscope, not telescope, microscope. And this guy here, Robert Hooke, who was the first professional scientist, he was in the Royal Society of London at the same time that Sir Isaac Newton was. And to say that Sir Isaac Newton and Robert Hooke did not get along would be an understatement. They always were accusing each other of nasty, underhanded stealing of their works and so forth. But they were both incredibly amazing people. And Robert Hooke built a really fancy microscope, and he started looking at all kinds of things under the microscope. And he made a book called Micrographia. And this is the first really popular, what you call a coffee table book. And it sold huge amounts. And in those days, up until really only about 60 years ago, if you were a microscopist, you had to be an artist because you had to draw what you saw. Cameras weren't hooked up to microscopes until about the 40s, the 1940s or 50s. And they didn't become really popular until the 50s or 60s. So you had to be able to draw what you're looking at. So microscopists were always artists. And this guy was a great artist and a microscopist. And he would draw these things he'd seen that nobody had ever seen before. So this is a flea. Now everybody had fleas in their dogs and cats, but they had no idea that they had beards and they looked like this. And he drew pictures of eyes of bees. And he opened people's mind to the idea that there was a microscopic world. Nobody knew there was a microscopic world. We look out there and we knew there were stars. We didn't know anything about them, but we knew they were there. But nobody had any idea that you could drill down and find an entire other universe in little tiny things until Robert Hooke came around and showed that to people. Then Anton von Leeuwenhoek saw Robert Hooke's work, and he got very excited about making a microscope. And he took a totally different approach about making the microscope. The microscope he made, he didn't make a big fancy one with multiple lenses. He went back to nature's original idea that a drop of water was the first lens that was ever created. So he was able to make these beautiful round lenses and just cut off the front right at the right place. And he inserted it right in this little holder right there. So that's that eye drop lens right there. And there was a screw and a needle, and he'd put the sample on the needle, and this screw here would focus in and out to allow you to focus. So he'd hold it up to his eye, and he'd put his eye right in the back of this thing. Right in the back of that thing. And with that little microscope, he discovered blood cells. People had no idea that, they thought blood was a liquid. Actually, it's filled with cells, red cells and white cells. He discovered spermatozoa. Can you imagine ejaculating and looking at these little things running around like crazy? I mean, that had to be a trip. I mean, that must have pretty much blown his mind. He discovered microorganisms. He discovered little microorganisms. Nobody ever knew there were protozoa and bacteria and all that stuff. Sorry, not bacteria, but protozoa. So he opened the world to all this incredible scientific stuff that existed. The crazy thing is, for the next 250 years, nobody used a microscope for science purposes. And that's kind of bizarre when you think about it. Leeuwenhoek's work was done, and it just that was it. Microscopes, they made lots of microscopes, but they were parlor microscopes. And rich people would buy these microscopes, and that was a form of entertainment. That's before they had movies and TV, and so you'd have a luncheon in the garden, and you'd bring out your microscope, and you'd look at flowers and sand and bugs and all kinds of interesting stuff, and that was their entertainment. It wasn't until about the middle of the 1800s that microscopes became instruments of science, mainly because of this guy here, Ernst Haeckel, also because of people like Louis Pasteur, which I'll get to in a minute. But Ernst Haeckel, so he was a biologist and an artist. And he was fascinated with marine life. And he was invited, this is the time of the age of transportation and communications. They were laying the transatlantic cable. They laid a cable, big cable of wires from Europe to America. And it took them three attempts. They finally did do it. And the transatlantic cable connected by telegraph Europe to America. And he was on the expedition of the Challenger, the HMS Challenger for England, to help chart the bottom of the ocean. Because what they do when they were laying these cables, they would dredge up what was at the bottom of the ocean and take a look at it. So he was on these expeditions with his microscope, and he discovered thousands of species of diatoms and all kinds of interesting marine life. And he would draw them. He was a really good artist. But his drawings became more and more fanciful and more sort of sensual. So these are some of his drawings of what he found. And they became really sort of, I would say, almost erotic and beautiful. And he started making fine art out of this stuff. So it was sort of one of the first crosses between science and art was this guy, Ernst Haeckel. And he wrote a book called Art Forms in Nature. And if you get a chance, this book you can get in libraries. It's probably here in this library. Hopefully it is. If not, you should get it. Art Forms in Nature. So again, about the mid-19th century, these guys here used the microscope, Virchow, Schleiden, and Schwann, to come up with cell theory. So finally, the microscope was being used to really discover new things. And they discovered that all living organisms is cellular. And the idea is all life is cellular. The cell is the basic unit of all living creatures, so it doesn't matter if you're a bacteria or a mouse or an elephant or a fish or a jellyfish, you're made up of cells. Everything's made up of cells. And here's the killer. All cells come from the division of previous cells, which is pretty amazing. So take a look at this. So here's a dividing cell. Oh no. Is it going to play? Darn, it's not going to play. Too bad. So let me just go back. I'm going to explain what happened here. So this is a cell that's in the process of dividing. It's going to become two cells. That's the DNA. So at the point of division, the DNA doubles itself. It's got two full sets of DNA. The cell pulls apart, and now each daughter cell has a set of DNA. Now play that movie in reverse. Keep going back. So all of the cells in your body came from, originally, a sperm and an egg from your parents, and they started to divide into what you are now today. And their cells from your parents, they did the same from their parents to their parents, and it keeps going back. And it doesn't only go back to apes, it goes back to tree shrews, and it goes back to, all the way back to bacteria. There are cells, there is DNA in our bodies that are from bacteria and from many of the organisms that we evolved from over the last 3 billion years. So here's a cell. Cells all have jobs. Those are cells taken from the heart. So when I was doing my PhD, I did a lot of cell culture. And you could take a little bit of a cell and put it in a dish and photograph it. These cells are from the heart, and that's just what they do. That's their job. And if you put them in a dish, they'll just sit there and beat like that. And our hearts will do that till the moment we die. So I want to tell you a little bit about inventing microscopes. I've had the great good fortune to invent a bunch of microscopes. I have 20 patents right now for microscopes. I started doing this in the 1990s. So this is me when in my first year of graduate school. I started graduate school late. I was about 33 years old when I started graduate school. And I sort of gravitated toward using the microscope as my tool of investigation. And it's funny because people told me, oh, don't use microscopes. Everybody's seen everything in a microscope already. Now, 40 years later, the microscopes that exist today are so mind-blowing. They can see atoms. They can know exactly what molecules you're looking at. They can do the most amazing things. It's changed. It's completely changed medical science. It's created what we call nanotechnology. So at the time, I was being told that everything has been seen. You know, I didn't take that too seriously, and I kept using microscopes. So when I moved there, I moved to London. I got a job doing some special effects on the Superman film, which was being filmed in London at that time. And I did the first shot of the movie that looks like you're landing on planet Krypton. So in those days, when you did special effects, we didn't have computers to do special effects. You had to think of an idea. You had to think of something you could actually film and make it look like something else. So what I did is I got human pancreatic, I was studying human pancreatic cancer cells at the time, and I was looking at them in tissue culture. And you grow these cells in tissue culture like a big monolayer of cells, and they each have these nuclei. And the nuclei, if you use a particular type of lighting from an angle, it makes the nuclei look like craters, like craters on a planet. So I set up this thing, and I made it, I got a scene that looked totally like, and I colored it red and blue. And then I set up this apparatus on the microscope with all these gears here, where I'd turn the stage and it looked like you were going like this and flying over the planet Krypton. So that was my first, oh, by the way, look at this cool camera. That was a Panavision camera I did it with. Now, one of the things about the Panavision camera is interesting because you take the picture and the nuclei are round, but these Panavision cameras, what they did is they took a regular 35 millimeter film and they had special lenses that compressed the image so everything looked long. They're what's called anisotropic lenses. The image, everybody looks tall and thin, but then the projector, when they project the movie, it spreads everything out. So you get this widescreen effect. That's how widescreen was done in those days. So it was perfect for me because when I took these pictures of the cells, and I didn't use the anamorphic lens, so now they spread out this image, and these round things became ellipses, and it really looked like you were at an angle flying over a planet. So then I went to USC and began doing research with embryos. Embryos are very, very difficult things to see in a microscope. And I found that no matter what microscope I used, and I had every microscope available, I couldn't get a good image of these embryos for the research I was doing. So I thought, I think it must be in the lighting, because one of the things I noticed about microscopes that was really, really lacking was the lighting. If you look at a microscope, it's one light going straight up. Well, I came to science as a photographer, and I know that as a photographer, you never use one light straight on. I mean, a photographer gets key light, fill light, backlight. It highlights things, you create shadows, and it lets you see more that way. And I thought, wait, we could change the lighting regime on a microscope, and what would happen? It turned out it did all kinds of incredible things. By bringing in light from an angle, you would create highlights and shadows, so it created contrast for things that were otherwise pretty much transparent and invisible. Also, it increased not only the contrast, but increased the depth of field, so you get more in focus. And it also increased the resolution. So I came up with this idea of multiple oblique illumination. Rather than having one light, in this case there were 4 lights hitting a 4-sided pyramid, and you could turn on and off any of these 4 lights. You could use 1, 2, 3, and 4, and they had different effects. So one thing I noticed when I used light at this angle, I took a picture and I noticed it looked like you tilted the specimen. Even though the specimen was flat on the stage, it looked like you tilted it. And then if you put light from this angle, it looked like you tilted it this way. And I thought, wait a minute, if you could bring both those images to the left and right eye, you'd see that in stereo, because stereo 3D is seeing 2 tilted views at the same time. So our eyes only see 2D, but each is from a different angle. We're each seeing the same scene, each eye is seeing the same scene from a different angle, slightly tilted angle. And that tilt gives us the 3-dimensional perception that our brain creates. So I realized you could do that on a microscope, and not only do it, but at the same time improve the resolution, the contrast, and the depth of field, and get 3D. So that was my first invention. So one of the things about the resolution is interesting. I'll do this quickly because it's maybe too much science. But if you were to take this head off, of here and just take the back of this lens and put it, and you're in a dark room and you put a piece of paper and you have these are muscle cells here. What you'd see is this diffraction pattern. So what happens is the lens creates a diffraction pattern of the scene. And then if you take that piece of paper and slowly move it up until it comes to focus, that's what you see. It transforms itself. This image transforms itself into this image. And it became codified by a guy named Fourier, who was a great French mathematician, called the Fourier transform. And you can do this mathematically. And by knowing these Fourier transforms, you can do all kinds of things with images. It's a very, very powerful imaging manipulation technique. So the deal is this guy from Zeiss, Ernst Abbe, about the end of the 1900s, realized the resolution of the light microscope is limited. You can only see things about half the wavelength of light, and the wavelength of light about a half a micron. Now, a micron is a millionth of a meter, so you can see about half that. But anything smaller becomes invisible. And the reason is this. He says, in order to make an image, you have to have a diffraction pattern. And in order to have the diffraction pattern, you have to at least have the 0 order and one of the first orders. This is the first order. That's the second order diffraction wavelet. So when these markings, I don't know, can you see these lines here? Those lines, those spacings are represented by these spacings here. As the spacings get smaller, so this is a diatom that's just at the limited resolution of a light microscope. If these dots were any smaller, you couldn't see them, they would disappear. But just at that limit, what you're getting is the 0 order and the first order here. So as the markings on the object get smaller, the diffraction pattern gets bigger. And with the diffraction pattern, you lose it, you lose resolution. So I thought, well, what if you just use oblique illumination rather than, so here, this is a little cartoon of that last one. You have the 0 order and the first order. So if you put light, if you have something that's really, really small like this, you just get the 0 order and the first order is outside the lens, you see no image. But if you block off half of the illumination and use this oblique illumination, you could put 0 order on one side, the first order on the other, and now you've improved the resolution of a light microscope, and it gives you this tilted potential 3D view. So here's, for example, you're using light from the right side. So this, by the way, is a diatom that is beyond the limit of resolution. You can't see, with a regular microscope with straight-on light, you can't see these markings. But if you use light from the right side, you see these markings. If you use light from the top, you see those markings. And if you use multiple oblique illumination from both angles like this, you see the markings. All of a sudden, you've increased the resolution of the light microscope. Also, I realized that if you take a lens and you cut it in half, not literally cut it in half, but you block out half of it, if you do it in the right place in the optical system, you still see a perfectly good image of that specimen, but you see it from a tilted angle. So I realized the same thing happens with viewing a specimen as does with illuminating a specimen. You can take a lens, and I figured out a way to divide the eyes so the one eye sees the right half of the lens, the other sees the left half of the lens, and there you have stereo. So this is my first microscope that I invented, the Edge microscope in early 1990s. This was the apparatus that split the left and right eyes. So you had a camera with a left eye taking a left picture and a right picture. This is a camera that took a mono picture, both. And the multiple oblique lighting was down in here, and you could control any of the lights that you wanted. And it was a super cool microscope. You'd look in it and you'd see it in 3D. Then I realized also that if you took lighting and you put this sort of wedge of light on a motor and rotated it, stereo allows you to look at 2 angles at a time. But by rotating this lighting, it would make the object go like this, and you're interrogating it from 360 degrees from all angles. So I made these illuminating condensers that allowed you to do that, and that's what it could do. So that's a single brain cell from the cerebellum of the brain, and now you can see it in 3D without having to have stereo, without having to have any special apparatus. You can see the foreground from the background. So this is a way to allow you to see 3D without any special apparatus. So my latest microscope, this is my latest baby here, and this is the digital version of the microscope. It focuses at many levels, puts everything into focus, turns it into 3D. It's got the multiple oblique lighting. It's got digital, all kinds of incredible digital stuff it does. And the design of this microscope, I want to go back to Ted Tanaka, who was my partner, the architect from Environmental Communications. He did this design of my microscope. So that's why it looks kind of a little bit architectural. So now what happens in the microscope, because the big problem with microscopes is depth of field. Now, any of you here are photographers. You know that if you ever do close-up photography, the problem is lack of depth of field. You have one little part that's in focus, and everything in the background and the foreground goes out of focus. Well, in a microscope, that's like that's like even way more. It really happens badly. So this is close up on the stem of a water hyacinth, little part of a water hyacinth. So I took 32 images. This is the 6th image, and you can see there we're in focus right there. On the next image, this is the 11th image, and we're in focus right there. This is the 16th image. We're in focus up here. That's the 21st image. Now, if you put all 32 images together, now all of a sudden you can actually see what's there. So this is what modern microscopes do now. Modern microscopes take multiple pictures, and computers put them together. Computers figure out how to get rid of the out-of-focus information, put all the in-focus information together, and you really see what's there. So it's a real breakthrough. And this is the software that I created. This is just a piece of paper. You think a piece of paper as flat, but it's really in a microscope. Everything is 3D unless you're looking at sheets of atoms. So 12 years ago, I had the great good fortune to decide to retire and move to Maui. That did not go so well, the retirement part. Whoops. The retirement part didn't go so well. But I noticed that a couple of things that when I got there, I first started doing some substitute teaching at Kamehameha Schools, which is an all-Hawaiian school. And I had me and my microscope and nothing to do, and there was a lot of sand around and there were a lot of flowers around. So I started taking pictures of sand and flowers and other things. This right here. And so what I found is the microscope is a great place to do art because everything you look at is a surprise. Everything is abstract. It's nothing you could have possibly imagined that it looks like when you look through a microscope. I'm going to show, this is sugar crystallizing. So this was the cover slip, the edge of the cover slip. So this is the crystals of sugar that were crystallizing, and then here they're crystallizing in 3 dimensions. This is more kind of flattened. I want to show you a little movie. Oh no, sorry. This is, I started taking pictures of wine crystals. So if any of you have ever looked at a cup of wine or a glass of wine the next morning, you notice there's some grungy stuff at the bottom, some sort of cruddy stuff at the bottom. Those are wine crystals. And I started taking pictures of them. And every type of wine had a different type of crystal. This is Merlot wine crystals. This is Beaujolais wine crystals. This is Zinfandel wine crystals. Here's a little movie. So you can also do a lot of time lapse. I've done a lot of time lapse in my life because that lets you see hidden patterns that you could never see otherwise. So this is a little movie that my son Dorje, Jason Greenberg, and I made. So if any of you ever made rock candy when you were small, that's what's actually happening microscopically. Making rock candy, so what I did there is I took some supersaturated solution of sugar. In other words, you boil some water, dump some sugar in it, as much as you can, until it won't hold any more sugar, and then let it cool, and it begins to crystallize. And if you put a string in it, it makes rock candy. Some of you may have done that when you were younger. So I've found that when I look at things through the microscope, just ordinary things, I found that ordinary things are really, truly extraordinary. Just things like sand. You wouldn't really believe what sand looks like. So if you look at, this is a beach in Hawaii, McKenna Beach. If you look at it, it looks like sand. You look a little closer at the same thing, and you look a little closer at the same thing, all of a sudden you see, wow, there's some stuff happening here. What's this here? That's like a sea urchin spine, a little bit rolling around in the surf for ages. That's a little bit of one that's been crystallized. There's a little bit of the volcano. It's a tiny grain of sand. Look, it's got 1, 2, 3, 4 different types of minerals all squeezed together into a little grain of sand. There's a little tube worm down here, lots of shell fragments. So sand is amazing. This is the first sand I looked at when I got to Hawaii. This was from Lahaina. And I thought, oh my goodness, I thought sand was little brown rocks. And look at this stuff. Here's a sponge spicule here. And these are this olivine and some crystalline glass. And there's a fragment of a shell. And there's a sea urchin spine and a tube worm and microshells. Look, these are tiny, tiny, tiny shells that are in perfect shape. Some of these are foraminifera. This is a little bit of from the volcano. And look inside this little pocket, is a little crystal that crystallized. There's a whole world of stuff, even in a single grain of sand. So I realized, gee, this is a really good place to do art. So I got a set of acupuncture needles, and I started going through sand and finding the really cool little grains of sand and making these little designs and making pictures of them. So this is one of my pictures of grains of sand from Lahaina. And I did a book on sand, published by Voyager Press in 2009. And when I developed that book, NASA saw the book, and they realized they never got good pictures like I was taking using my 3D microscope from the sand from the Apollo missions. This is the second book I just wrote on sand, about sand last year. This has pictures of the moon sand in it. So I got the people from NASA came to Maui, brought me moon sand. Then I got a grant from NASA to take pictures of moon sand. I did a lot of talks and went to conferences and so forth. So I want to show you some examples of sand. This is sand from the Galapagos. Whoops. These are not playing as I had hoped they were. Sorry, these are supposed to loop. That's from the Galapagos Islands. And again, using this technique, you begin to see the 3D, using this rotating technique. These are grains of sand from Skeleton Beach in Namibia. This is on the beach, up in the mountains, they do mining of gems there. They call it Skeleton Beach because there's a lot of diamond mining there, and you can actually find diamonds on the beach. And they're human skeletons they find there because it's in the middle of nowhere and people kill each other for these things. This is obsidian sand from Santorino in Italy. This is from a green sand beach in Hawaii at South Point, made of olivine. This is the famous star sand from Okinawa. These are little tiny microorganisms called foraminifera. And they were living at one time, and when they die, they leave behind these little grains of sand, and they're star-shaped, and they're fabulous. And also there's these little itsy-bitsy tiny shells in there, and there's a sea urchin spine and other kinds of foraminifera. These beautiful stars with embedded with little pearl shapes. So this is a grain of sand from the moon, a little tiny itsy-bitsy grain of sand from the moon. This grain of sand was formed about 4 billion years ago, yet it has not eroded away. If this were on Earth, it would erode away well, depend how hard. That grain of sand would probably take about 1,000 years to totally erode away. But it's been about 4 billion years since that was formed. Its original structure is still there. The only thing that's happened is you see these lines here, and these were caused by high energy impacts from probably cosmic rays or something like that, some bombardments from outer space. There's a little grain of sand that was the tip of a cone shell, and the little tip broke off, and it just rolled around in the surf for ages and became transparent and translucent and just beautiful. Some grains of sand are just look like phalluses. Every grain of sand is different. There aren't two grains of sand alike in the universe. Every one is different. Now imagine if every grain of sand is different, imagine how unique each of us is. And that's something we should remember. And the way to find out your uniqueness is to discover your point of view. That's why photography allows you to do that. Art lets you do that. So people ask me, is that what sand really looks like? And what I tell people is there's no such thing as what something really looks like. Because it all depends how you look at it. It depends what light you look at it in, what tools you look at it with. So there's no such thing as that's what it really looks like. We don't have that kind of perception, the ability to perceive the universe that way or to perceive the world around us. So that's some sand grains using reflected light. Here's the exact same sand grains using transmitted light. So they look pretty different. And more than just the lighting, it's the type of energy that you look at it. So for example, although this is called light, this is through a light microscope. This is a grain of sand from the moon. Lots of grains of sand from the moon have holes in the middle of it because the moon is always being bombarded by micrometeorites. And they put holes in sand, and these are called ring agglutinates. And these grains of sand are really, really interesting. And if you look here, you can see with a light microscope, you can see there's some orange color here. There's some dark, some lighter orange, some dark colors. You can see what's transparent. You can see what's translucent. You can see what's opaque. Now here's the exact same grain of sand taken with very high energy electrons using an electron microscope. In this case, you can see a lot of the detail of the surface, but you don't see what you can see with a light microscope. You see different information. And here's again the same grain of sand looking with an X-ray microscope. And now you can see right through the inside of it. So what does this really look like? What science does, it looks at things from multiple points of view. And after you're looking from as many points of view as you can, then you come up with a model of what it is you're actually looking at. That's what science is all about. And not only when we say we look at something, we only can look at things. Human beings can only look at the visible radiation. So there's radiation in the universe of the whole spectrum of electromagnetic radiation. We can see this little tiny band in the middle, from 400 nanometers to about 700 nanometers, from blue to red. And on the higher energy side, there's ultraviolet, and the longer side is infrared. So if infrared is, we're all radiating energy right now in the infrared. We're lighting up. We actually light up. If I turned off all the lights here, now we don't have the ability to see infrared, but if I turned off all the lights and I had an infrared camera, you'd see, and you guys have probably seen this, we're actually radiating. Those colors, we're literally radiating energy in the infrared. So birds and some birds and insects, they have the ability to see ultraviolet. So when they see flowers, when we look at a flower that looks like this, they may look like this. There's a totally different pattern. So this picture was taken with infrared film, with ultraviolet film. And you can see there's a different pattern when you use ultraviolet film than if you use visible light, what we see. Same thing with this flower, same thing with this flower. So insects see flowers even more beautifully than we see them. There are more patterns and more to see, and what they see is the microscopic world. So flowers evolved to show insects the microscopic reality of what they are. And that's much more beautiful than what we actually see, and I'm going to show you some of this. Also, by the way, I developed what I call the macroscope. This is the lower power version of the microscope I developed. It moves up and down automatically. You put a macro lens, a camera with a macro lens, and you take multiple pictures, and then my software puts it all together into one in-focus image. And that can let you do stuff with macro lens photography you could never do before. So this is a picture of a hibiscus flower. Now, the hibiscus flower is the one with this long, stamen and pistil coming out. We're looking straight down it, and that's about 4 inches deep, and it's all in focus. The whole thing is in focus from the tip all the way down to the bottom. This is about 40 pictures put together. So you see the whole thing all in focus. And this is like looking at this here, but straight down. So I want to tell you a little about the birds and the bees now. We'll get onto this subject finally. So the hibiscus flower is the most outrageously erotic flower that there is. I mean, its display, its sexual display is really in your face. It's just really out there. So this is the female part. This is the stigma, and this is the male part. These are the stamen. Most plants are hermaphrodites, but they don't. Most plants cannot pollinate themselves, and that's for good reason. So if plants were able to pollinate themselves, there would be no diversity in the DNA of that organism. So plants that early on were able to pollinate themselves, they died out. Most of those died out because there was a great advantage for plants not to be able to pollinate themselves. So there are various mechanisms by which they cannot actually have sex with themselves. And one is that these mature at different rates. The maturation of this is not at the same time, or they have some biochemical markers that won't allow them to have sex with themselves. So what it's all about is it's the birds and the bees. The bees are there to collect the pollen. When I say bees, I mean all the other insects as well. And they go around and they collect pollen. For the bees, they're bringing it back to their hive, and they make the hive, and they collect also nectar. And the birds, what they do after the seeds are formed, the birds eat the seeds. They fly away miles away, and they poop the seeds out, and now that plant is reestablished miles at a distance. So that's what sex is about, and that's why they call it the birds and the bees. So if you look close in, if you look close in right in this area right there with a microscope, this is how insects see it. It's a whole other world. It's filled with nectar. You know that plants can make their own food. They take carbon dioxide and water and sunlight and turn it into glucose. And this is what they make. They make nectar. And this is a little tiny bug, the insect that lives in this is smaller than you can see with your naked eye. This particular one lives in the hibiscus flowers. And it's like a Willy Wonka world to them. So these flowers not only smell good, they look good, and they taste good, and they provide food and housing for insects. So bees are busily moving around all day long, collecting this stuff, going crazy, doing their thing. And if you look at a microscope with the legs of a bee, sorry, I took that out, I guess. A leg of a bee is they grow hairs, and they pack the pollen in between the hairs so they can take it back to the hive. Now this is what one of these little yellow dots looks like when you look at a hibiscus flower and you see a yellow dot and you look with a microscope. It's got hundreds and hundreds of grains of pollen, and the pollen is sticky and spiky. And the idea is that when the insects are attracted, they get caught up, and the pollen gets caught in their arms and their backs and their legs, and they move from flower to flower, and they can pollinate other flowers. So that's what it's all about. So this is one pollen grain that's been embedded into the top of one of these stigma. So this is the female part, and that's the male part. And what happens is this will merge down into the flower. Let me see if I'm showing this. Yeah, so it'll come all the way down the flower to the inside of this flower, down what's called the pollen tube. I've cut this flower open. You can see the middle. That pollen comes, and it's got two sperm cells, the pollen. And it comes all the way down, and this is the ovule, this is the ovary, and each of these are the ovules, and they get pollinated, or they get germinated by the pollen. And what happens is each of these turns into a seed, and this turns into the fruit. So that's what fruit is. The fruit is when it gets pollinated, that becomes the fruit with the seeds inside. Now, the strawberry was one of the very few fruits that has seeds on the outside. That was the first picture I showed you. Now, here's a seed through a microscope, just the beginning of a seed being formed in a cucumber. Here I did a little thing with lighting in the microscope, and I told you my thing is to create sort of unique lighting. So I had some oblique lighting coming from reflected light, and then I got some sort of goldish yellow light coming up from the bottom to give this sort of lacy silver gold look to it. So now I want to show you a little bit. Let me see how I'm doing on time here. Okay, I better move it along. I'm going to go through these quickly then. So I've been working on a book called Florotica, revealing the sensuality of the micro world. And I just want to show you, I'm just going to go through and show you what some of these pictures look like, because otherwise we're not going to have time for questions. So this is the shell ginger using a macro lens, and if you get in closer, look at these wonderful patterns. And when you look at it with a microscope, it's just juicy. It's really nice. This is a water hyacinth, and when you look in closer in the middle there, that's where the business part is, the stamen and the pistil. I mean, look how inviting that is for a little insect. You know, we can't see that unless you happen to have a microscope and a lot of patience to make a picture like this. Here's a little tiny, a really small orchid. It's about an inch or two. And if you look at this area, right, we're going to look at a couple. We're going to look at this area here, and then we're going to look at that area up there in the microscope. Each little dot here is a cell. And the DNA in that organism determines where every cell is going to be, what its color is, how long it's going to live for, when it's going to shrivel up, and so forth. This is a Hong Kong orchid. This is the female part. This is the male part. By the way, interesting thing in flowers, usually the female part looks like a phallus and the male part looks like a vagina. It's kind of usually the other way around. I mean, it's the other way around, which is strange visually. So this is the female part of the Hong Kong orchid tree. It's a beautiful tree. It has a slight fragrance to it. There's the male part. Here's a little coral tree flower, and that's what the male part looks like little. And there's the female. There's the female part. Hard to believe that's the female part. That's the male part. This is my favorite flower. This is the hibiscus, and it's the state flower of Hawaii. They're just fantastic. Here's a little visitor up here, and that's looking at the female part up under the skirt, so to speak. That's from the side view. That's the part that receives. So this is the female part, the receptive. This is the receptive female part. These are the stamen that have the grains of pollen. It was supposed to be a little smoother than that. Anyway, you can see that the microscopic world is completely different when you get up with a microscope. This is the stamen before it's opened up. These are, they're closed, and the pollen are being developing inside of these structures right here. That's before it's opened up. Now it opens and releases its many grains of pollen. I want to show you what some of the petals look like. That's the petal of that flower. Each little dot there is a cell. Here's a red hibiscus, and that has a very different petal. Each petal here have these trichomes. And this is part of the sensory apparatus and what gives the aroma to some flowers. Here's an nasturtium, which is an edible flower. That's some of the pollen and the petal. Each little brick-like structure here is a cell. Each of those is a cell, so we're in very close. Here's one of the really beautiful local flowers around where I live. Has a wonderful fragrance. And that's the male part. Sorry, these are jumpy, but that's how it goes. It was supposed to be smooth. That's the petal of that flower. Here's a dietes flower. So I wrote another book called A Bug's Eye View of Flowers, because that's really what this is. I realized that bugs see flowers totally different than we do. So this is like the bug is, this bug here is looking at this flower here, and this is what it would see. It's seeing like what a microscope would show you. And there's that same flower. And there's a microscopic view of it. And that's a protea, and that's the male part. Quite beautiful. This is a cleodendron. And this is such a beautiful stigma. Look at that. I'll show you a little closer up in a microscope. A lot of this stuff is really sort of, really has this sort of innate sensuality to it. That's the male part of that flower. Here's a jacaranda, and the blossoms, they start closed and then they open up. And that's what it looks like in a microscope when you look up close. And here's a white ginger. And here the male and the female part are right next to each other. That's the female part, that's the male part. And that's the leaf. That's the petal. That's this petal here. I was really surprised. This white petal, when you look in a microscope, that's what it looks like. I've never seen a petal that looked like that. It's really, really beautiful and different and unique looking. That's a wonderful tree that we have in this is the female part just here. Let's see, that's it close up. That's the same flower again, and that's the petal of that flower. And here's just some shots of petals, just the sort of texture. This is sort of wonderful, sensual texture of petals when you look through the microscope. So here's this little orchid. So this design of this orchid is totally determined by the DNA in each cell of that flower. Each cell, like if we look at this little area right here, this little tiny tip, look at that through a microscope. This is what it looks like. Each dot here is a cell. Each cell knows exactly where it should be, exactly what color it should be. It knows that this should be yellow right here, and that should be red, and these should be white, and they should stop there, and that's all coded in the DNA. And here's the petal of a tiger lily through a microscope. Decidedly erotic. This is impatiens flower. It's the male part. That's the female part. The stigma. The receptive. Here's a really strange flower. The flower grows in the middle of the leaf. I'd never seen that before. And the little flower has a condom on it, which is pretty interesting. Leaves are where it's all about, what it's all about. The leaf is where the action happens in a flower, not the sexual action, it's where the energy comes from. So the leaf takes in carbon dioxide and sunlight and water, and it makes sugar. And in order to do that, it has to have little openings called stomata. You can't quite see them there, but if you go closer, you can see these little things that look like little mouths. And they're two little cells that open and close, and they open up to bring in carbon dioxide and then close down, and then when they're ready to expel oxygen, which is the byproduct of photosynthesis, they open up and let oxygen out and then close up again. If they didn't do that, leaves would dry out. So this was an important evolutionary development in plants. Here's this is the leaf of a basil. Look, it's just loaded with these little dots of oil, wonderful little oil dots. So this is where you get the fragrance and the oil from basil. There's closer up. It's a gland, and that gland makes the oil that we know as basil. And lots of these aromatic plants that we use in cooking look like this when you look at the flower. So I want to close up with one more thing here, two more things. I started out showing you, I want to make 2 points. One is that instruments of science can be really important instruments of art. They can be great tools of art. This is the picture taken by NASA about a year ago, maybe 2 years ago, I forget, a year or two ago. It flew around the back side of Saturn and got right Saturn exactly eclipsing the sun. So this is a Saturn eclipsing the sun. One of the most beautiful, first of all, Saturn is one of the most beautiful planets in the universe. And secondly, this picture of it is probably the most beautiful picture. Now, I don't know if you can see it. Can you see that dot right there? Let's see. It's not showing up, huh? There's a little dot right there. There's a little dot right there. That's the Earth. That's the whole Earth. And there's another one right there. I'm sorry you can't see it on the screen. That's Mars. So now our view of the whole Earth and our world today is a little tiny dot floating in space, barely, barely visible. In fact, in your case, it's not visible. Sorry, you can't see it there. I want to end up with one more thing about the micro world. We have microscopes today that can see atoms. Now, I know you've been told that everything is made of atoms.
This is just an ordinary
but I want to show you it's actually really true.
One flash, and the motor starts. But the white, isolating sheath is far from ordinary. It's made of alumina, a sophisticated ceramic capable of resisting extremely high temperatures. Tiles made of this ceramic form the protective casing on the space shuttle. We can now see a tiny chip, which was invisible just a few moments ago. The fault appears to have devastated the smooth surface, creating a chaotic landscape of sharp ridges. Terraces materialize on the slopes of this mountainous structure. They're the steps of a staircase resulting from breaks in the atomic structure of the alumina. A multitude of these tiny breaks, each one perpendicular to the other, gives this angular aspect to the surface of the ceramic. As we look into one of these steps, others, even smaller, are revealed. Deeper inside, we discover a microstructure, more detailed still, made up of minute grains of alumina. The diagonal line crossing the screen joins two of these grains, welding them together and giving the structure strength. The fewer impurities there are in these joins, the stronger the ceramic. Even under such high magnification, this join appears clean and neat. Moving closer, the surface is bloated and cracked. Electrons emitted by the microscope have almost punctured the matter. By luck, the two grains of alumina are positioned differently. On the left, the atoms are seen flat, the vertical columns of atoms giving this striated effect. In contrast, the grain on the right shows up as a cross-section, each white point representing the peak of one of these columns.
Each dot there is a single atom.
Alumina is known by another name, aluminium oxide. At this point, among a uniform mesh of oxygen and aluminium atoms, the microscope reaches its limits.
Now, all of these movies, when you do this, end up the same. As long as you're taking a picture of something solid, as opposed to a gas or liquid, they'll all end up with this matrix of individual atoms strung together in a beautiful, beautiful design. So I want to end with one little, it was really, I would say that William Blake really said it best. To see a world in a grain of sand and a heaven in a wildflower. Hold infinity in the palm of your hand and eternity in an hour. Thank you.